Okay, welcome everyone. Welcome to our Cup of Tea event. This is our event which is intended to bring the Canadian retinoblastoma community together to talk about retinoblastoma research in plain language. Discussions center around articles which members of our Canadian Retinoblastoma Research Advisory Board, or CRAB, have created plain language summaries for. You don't need a research background to participate. Your questions, perspectives, and experiences are what make these conversations meaningful. So please join our discussion after our article presentation. Our past Cup of Tea recordings are available on our YouTube channel and Spotify under the channel's Retinoblastoma Research. And you can contact the Retinoblastoma Research Advisory Board about any information shared today. If you have any questions about anything, retinoblastoma.research@sickkids.ca. So we're here on the January 20th presentation. My name is Ivana Rostevsky, and I'm the parent in research who's hosting this event. Our agenda for today is to go over some announcements. I'll introduce the cup of tea, and then we'll go right into the research article, which will start with our patient partner, and then our research partner will present, and then we will go into our community discussion where everybody can participate. So first, I want to encourage you all to sign up to our retinoblastoma research community. If you have not done so already, I think most of you have, but not all. I just want to remind everyone that this is a great space to learn about opportunities to partner in research and receive a quarterly newsletter, which may include information about meetings and events, education and training, and other opportunities. So it's the best way to stay informed. And if you're wondering maybe if you should or should not join, yes, everybody can join. Anybody who has lived experience or professional experience with retinoblastoma can join. You can go on our website, rbcanadaresearch.com, and I have a little screenshot of what the top bar looks like. The one that says RB Research Community is what you click on, and it will direct you straight to where to sign up. So we have some opportunity. Yes.
I'm sorry, Ivana, may I add something to that really quickly?
Yes.
Just if there's anybody who isn't sure if they're a part of the research community, you can always reach out to retinoblastoma.sickkids or retinoblastoma.research@sickkids.ca. I'll enter that into the chat. If you think you might have an old email, we can also always update that for you.
Oh, that's really helpful. Yes. Thank you. So we have many opportunities today to share about partnering in research. I am always looking for cup of tea partners. I'm looking for somebody with lived experience and non-lived experience to partner with. So if you send an email to the Retinoblastoma Research mailbox, I can contact you and share more information about that opportunity and what it entails. I have some pictures here on my slide about past participants and them presenting during a cup of tea event. And you can see they're all smiling. So it's a pretty good experience, I'd say. I am also gathering input about future cup of tea topics. So this is a survey we have, which also went out in our January newsletter for those who are part of the retinoblastoma research community. If you could please take the time after our meeting today to fill it out and provide some feedback on what you want to learn more about and for future cup of tea topics. And we will use this information to set the next round of topics for our cup of tea events. Also, we have our Patient Advocate Committee, or PAC, for the Rare Pediatric Eye Cancer, or RPEC Biobank. They're meeting monthly with lots of patient engagement activities. And anybody who's interested, also email retinoblastoma.research@sickkids.ca for more information. There's a community event to participate in research. The International Retinoblastoma Consortium has some roles in leadership and advisory for people with lived experience to support DEPICT Health. There's an applications deadline, which is February 20th. And there's some information online that on their website that they have to provide more information and to apply. And that link's being shared, was shared also in the chat. For our future events that you can look forward to, we have the March Cup of Tea. The topic will be decided from the survey that's being completed right now from the newsletter that was sent out and that I just mentioned. The date would be March 17th at 8 p.m. Eastern Time. The event after that would be the RB Research Symposium happening on April 18th and 19th. It's a hybrid event, so visit our website for some more information, the location, to sign up, all the info is there. And following that would be our June Cup of Tea. Again, topic will be determined closer to the date, and that will be on June 16th at 8 p.m. We also have one Retinoblastoma World, also happening hybrid and in person in San Antonio, Texas, September 12th to the 14th. And their information is on their website in the chat about the event and how to register. So lots of stuff happening. Put this on your calendars and please attend. Okay, so for today's event, we have patient partner and a non-patient partner who have created a plain language summary for the article, Deneddylation of ribosomal proteins promotes synergy between MLN 4924 and chemotherapy to elicit complete therapeutic responses. This is a great article to share today because we can bring something that's a bit more complex to people who don't have a science background and discuss it using more plain language with our two experts here. So I would like to welcome Dhruv Kohli, who's a retinoblastoma survivor and is in his third year at McMaster University studying integrated biomedical engineering and health sciences. He's currently working with Dr. Malapatna to help create a hyper-realistic 3D eye model of retinoblastoma using his own anatomy. And Rod Bremner, who is a researcher at the Linfield Tenenbaum Research Institute at Mount Sinai Hospital. He began his career by developing models of retinoblastoma, including inherited forms of the disease. Although his laboratory now studies cancers more broadly, his work continues to be strongly informed by insights gained from retinoblastoma. So welcome, Dhruv.
Awesome. Thank you, Ivana, for the warm introduction. So, hi, everyone. It's great to see a lot of familiar faces. My name is Dhruv. I am in my third year of integrated biomedical engineering and health sciences at McMaster. I'm trying to become a physician, applying to med school in the next coming years, in hopes of becoming a physician. After all of the amazing care that I've received at Sick Kids, I hope to provide that to my own patients someday in the future. So just in terms of my story, so I was diagnosed with retinoblastoma around the age of 6 months old. And the way I was diagnosed is pretty typical. So we tell parents now to take pictures of their kids when they're younger, and to look for that white glare. Well, that's exactly what happened to me. My mom was, as a new mother would, taking pictures of her kids. And she saw a white glare in my eye that was not seen in any other photos. She told my family members that she thinks something may be amiss, and people thought that nothing was wrong. So she brought me to a walk-in clinic. Actually, the first time, they thought nothing was wrong, but after getting a second opinion, it was found that I did have retinoblastoma. So I was referred to Sick Kids and I received prompt treatment. And yeah, I'm here today. So in terms of how I was treated with a combination of chemotherapy and cryotherapy. And that allowed me to retain both eyes. So I had bilateral retinoblastoma, more so in my right eye than my left eye. My left eye, I have full vision, thankfully, but in my right eye, I have limited vision. I still do have my peripheral vision, but it is somewhat limited. Awesome. So as I do progress through university and I've become more involved in the integrated biomedical engineering and health sciences, I became really interested in novel research regarding a multitude of diseases, not just retinoblastoma. And so one of my biggest interests is these new advances in medicine. So when I came across this article, what I really saw was a novel treatment to the disease that I was diagnosed with at a young age. And this is something that I deal with today. And it looked really promising. So that's what really piqued my interest. As we know, chemotherapy is particularly toxic. We'll get into it a little bit more. But reducing the usage of chemotherapeutics reduces the side effects that children have to face with retinoblastoma. And that's really what piqued my interest in this study. So, yeah, as I've mentioned, chemotherapeutics are toxic treatments and they're used for a lot of different cancers, and they work by killing dividing and growing cells. So, not only are those malignant tumors, those are the majority of the cells in the body. So, most of the cells in the body are dividing. So, we do like to target the malignant tumors, but also things like hair and other cells are also collateral. And because they can't discriminate necessarily between these cancer cells and these healthy cells, there are a bunch of side effects. So I have an image here on the right. This is from Medical News Today, and it shows a lot of the side effects that are associated with chemotherapy. So these can include pain, hair loss, mouth sores, trouble breathing, a weakened immune system, nausea, vomiting, constipation, diarrhea, bruising, bleeding, rashes, and neuropathy. So, one of the things that has changed since I was treated when I was a kid, and this was one of the things that I was really pleasantly surprised to see actually as I started digging into this a bit more. Intravitreal chemotherapy is becoming more standard of care. And this essentially works by injecting the chemotherapeutic directly into the vitreous humor of the eye. This is a little bit different than how I was treated. I was treated more systemically through a port in my chest. So this has less side effects systemically. There are still some local side effects in terms of the healthy tissue around the eye. And we still do need to use these high doses to destroy these tumor seeds because if we don't, then the tumor can come back and we're going to need to treat it again. So I've included an image here on the right, just showing some different routes of medicinal entry into the eye. Intravitreal route being the one I just talked about. The topical route is how you'd receive eye drops, and there's a couple other here's for your reference. And so now just getting a bit more into the study itself. So the drug we're going to be talking about is MLN-4924. You might hear it referred to as MLN as we talk about it a bit more, just because that is a long name to say. But it is a precision drug. So that's something that Rod will get into a little bit in the future. So it basically targets the tumor more accurately, and it reduces the damage to healthy tissue. And the great thing about this is that it not only reduces potential side effects, it can also be more accurate. It can be more accurate to that tumor. And so the way it does this, what they found in the paper is that it works by blocking Deneddylation. So this is a cellular process that these retinoblastoma tumor cells rely pretty heavily on. And they found that in combination with current standard of care chemotherapeutics such as topotecan, MLN is very effective. So this is really important because as a precision drug, we're able to use less of other chemotherapeutics, meaning that we can actually achieve similar or better outcomes than the current standard of care, with less toxicity to the surrounding tissue. So I'll pass it over to Rod now, who'll take you a bit more into the crux of the study.
Thanks, Dhruv. I want to thank Ivana for inviting us to talk and also thank her for clicking the slides. So I might ask you as well, Ivana, just to point to something because I guess you've got your pointer there. Anyway, Dhruv did a great job of introducing this idea of like traditional chemotherapy and targeted or so-called precision drugs. So a lot of the chemotherapeutic drugs are really toxic to dividing cells, and so they kill dividing cancer cells, but they can kill a lot of normal dividing cells too, and they can also have side effects on non-dividing cells as well, because a bit like an atom bomb here. So the atom bomb will, obviously blow up the thing you want to blow up, but it's going to have a lot of collateral damage. And these other therapies, these more specific therapies that are a bit more like this, the gun with the sight here, you can still get collateral damage from these kind of drugs, but it sort of tries to target an Achilles heel in the cancer. From all the molecular knowledge that we have about cancer over the last 30 or 40 years, we've begun to understand ways to really get at the weak points in cancer cells. And so this way you can kill the cancer cells more effectively, and as a bonus, potentially have fewer side effects. So on the next slide. So I just want to give you a couple of examples of precision drugs. Often the idea of a precision drug is that it actually treats a defect, a specific defect, a genetic defect in the cancer itself. So there's a type of leukemia. This is the first precision drug really in chronic myelogenous leukemia, which is a blood cancer, CML. And it's actually caused by the fusion of two genes together that should never be together, BCR and ABL. And the old chemotherapy, basically about 30% of patients would survive for 5 years. But this new drug, imatinib, which targets this fusion protein, BCR fused to ABL, nowadays 83% of patients survive 10 years. So that's a remarkable success story. And the next one is an example from breast cancer. It's less dramatic, but nonetheless, hundreds and hundreds of women's lives have been extended and saved because of this, because there's a huge number of women who get breast cancer. And so there's a type of breast cancer called the HER2 plus subtype. So HER2 is another protein. It's very important in breast cells. And some breast cancers have too much of this. And the drug that targets this is called trastuzumab. And in a trial called the HERA trial, the standard of care was chemo. The patients, 5,000 patients got chemo, they got radiation and surgery, which is the standard of care. And then afterwards, on the top there, some of the women got no further treatment, which is exactly what we've been treating this disease, the subtype of breast cancer with forever. And the 63% had could survive to 10 years. But those who were in addition given trastuzumab for 1 year, which targets HER2, which is this thing that's overrepresented in the cells, there's too much of it, 69% made it to 10 years. Now that's a 6% increase. So you can imagine that's hundreds of women in this trial who lived to the 10-year measuring point. And the next is looking at retinoblastoma. So retinoblastoma, we've used chemotherapy, and there's been success stories. Obviously, people like Druze have benefited from chemotherapy, and especially with intravitreal injection, things have gotten even better. And you can do it systemically, or you can inject it to an artery near the eye. And that's some of the names of the chemotherapeutic drugs there. Those are kind of the atom bomb type of drugs. And you can also inject into the eye another kind of atom bomb drug called melphalan, or you can use topotecan as well, and these are quite successful. Actually, topotecan is less toxic than melphalan, and it's preferred. But there's still risks of side effects from these drugs. And so the use of sort of more modern precision drugs, topotecan was actually identified in the 80s. And it came from, it's actually a derivative of another drug that was identified in the 50s from plants. But the more modern drugs, none of these have been applied, for example, in CML or breast cancer. Those kind of drugs, the modern ones, have not been applied really in retinoblastoma. Okay, so this MLN4924, what a horrible name. Drug companies then change the name and give it an even more horrible name, pevonedistat. Doesn't really matter. We'll just call it MLN to be simple. And this is a precision drug. It really has a very specific target. It inhibits, as Dhruv said, something called Deneddylation of select proteins. What on earth is that? Okay, so you got this protein X, the blue protein X there. And there's an enzyme which nedylates the protein X, and it sticks on the NED8 protein, which is in red up there. So the NED8 protein actually gets chemically fused by this enzyme to protein X, and there's several of these proteins that get nedylated, and there's several proteins in the cell that need to be nedylated to function normally. And the drug, MLN4924, inhibits this enzyme. So that's what that little inhibitory sign is below the nedylating enzyme. That's what MLN4924 is designed to do. So it specifically targets this enzyme that does this job. And then if you just click the next part, So we came along, other people discovered this drug in 2010, and we showed in 2020 that this drug is a potential new therapy for retinoblastoma. I won't get into the specific reasons why we chose it. There's a very specific molecular reason why we chose it, but I don't have time to get into that. But we published this study in 2020, and this was work done by Arthur Aubrey. I was a graduate student in the lab at the time. Arthur would have been here tonight, but he lives in France, and it's about 2:30 a.m. in France, so we can forgive him for not showing up. And then, however, in that paper, we showed that while some retinoblastoma cells were really sensitive to the drug, single treatment with this drug, others were much more resistant. We needed much higher amounts of the drug to have an effect. And so we set out to deduce why that is. So in 2023, we published the paper we're talking about tonight. And again, that was Arthur that did this much more detailed study. And that's what I'm going to tell you about now. So on the next slide. Again, just to remind you, some retinoblastoma cells are really sensitive to this drug, but others are much more resistant. So why is that? So just to explain, each gene is basically a recipe for a protein. And some proteins are used by drugs. They help the drug to kill cells, like cancer cells. But other proteins hinder drug action. And so we screened through thousands of genes with a technique that I won't go into, but that's what we did. We just asked one by one in one experiment. We wanted to find which proteins does MLN activate to kill retinoblastoma cells, which ones are working with the drug to help us cure the cancer, and what about in resistant retinoblastoma cells? Do these show up in a resistant context? And so this is a horrible looking thing. Actually, this is actually from the paper. This is a picture of all the things we discovered. And you don't have to look at all those names and the blobs, but the blobs are all proteins, and the red ones are good ones. Those are the ones that are helping the drug to kill the cancer cells. And the blue ones are inhibiting the drug from killing the cancer cells. And the brighter the red, the more potent they are, and the brighter the blue, the more potent they are. And overall, these sensitive ones, you can see they've got lots of red guys. These are the good soldiers. And the blue ones are the kind of bad soldiers fighting against your drug. This is true of any drug. But then if we look now in resistant retinoblastoma, so if you click to the next one, this is what we found in resistant retinoblastoma. Hardly anything. So MLN engages a big army of killer soldiers, killer proteins and sensitive cells, but it's really pathetic in resistant cells. Hardly anything shows up, and that's why these cells are resistant. Okay, so the next slide. Before I go on to the next part, which is where we solved how to make this drug better in these resistant cells, I got to explain something to you called synergy, drug synergy. And this is where drug A has a partial or no effect, and drug B has partial or no effect. But when you combine them together, they have an amazingly strong effect. And so the combination is not just like A plus B, it's much more like A times B. And as an analogy, if you click the next. So if you imagine in a dam, if you have one hole in a dam, you've got one weakness, a minor effect, a little bit of leaking. But if you make two holes, something very catastrophic can happen. So that if you make these two holes, you really weaken the whole structure, and boom, the pressure of the water just breaks down the dam, and catastrophe, craziness. So that's a really graphic example of synergy at dams. And so the other great thing about synergy is that we can, because we can use much less of A and B to get this huge effect, we can also reduce the drug amounts and also reduce the toxic side effects. So there's a real push these days to try and identify synergistic drug combinations. So the next slide. We found, so when you're trying to develop a new treatment for a disease, you can't suddenly put a cancer patient, just on the new drug. You have to give them standard of care. Ethically, you must. And so ideally, if you can find a new drug, which is great, and it synergizes, with the standard of care, that would be really fantastic. So we tested it with melphalan and topotecan, and we found that it actually synergizes. It really kills the resistant cells very effectively. So these combinations are great. And so now we went back and we repeated the screen of thousands of genes to identify the proteins that are engaged either by MLN, TPT, or MLN plus TP in those resistant cells. When we do MLN alone, I showed you that before, there's hardly any soldiers that come out. When we do topotecan alone, it's a little bit better, but it's still kind of sad, right? Now, what happens when we combine them? Boom, there's all these soldiers suddenly appear. There's this big army of proteins that are engaged, killing the cancer cells. So all the red guys there. And what you'll notice, if you look on the left-hand side, so the left-hand side, they're talking to, the lines mean that they're talking to each other. And on the left-hand side, you'll see a lot of proteins that begin with the letters RP. And those are all related to each other. And so they're all kind of similar related proteins with similar functions. And they have a very important role in the cell, but in this case, we're finding that they're killing cancer cells. So what's going on? And they're also collaborating with all those chaps on the right. But the bottom line is that, there's this synergy. We can explain the synergy at the molecular level because we're basically getting all of these soldiers out of the barracks and into action. So if you go to the next slide, I'll show you what happens with these RP proteins. Why are they suddenly doing this new thing? They don't normally kill cells. They have another function completely in the cell, which is really important. Okay, so what we found was that Deneddylation ensures that these RP proteins go to the right address in a cell. So proteins are sent to specific places in cells to do a job. And the blue here, that's the nucleus. That's all the DNA in the nucleus. That's all your genes in a nucleus. Each one of those is a cell, and each one of those blue things is a nucleus inside the cell where all your genes are. And then if you look at the green now, below that, that's where RP is. This is a particular RP protein, RP11, and you can see there's a blob of it inside the nucleus. There's a little circle inside the nucleus, and then there's some outside the nucleus around in what's called the cytoplasm. Now, what's that thing inside the nucleus? If you look at the pink bit, that's something called the nucleolus. You don't have to know what that is, but it's a substructure inside. It's a particular address inside the nucleus that this RP11 protein is going to. Now, what happens when we treat with MLN? So if you click the next, there's a gigantic effect in the localization. Here's a nucleus of a retinoblastoma cell which has been treated with this MLN drug. And so there's the nucleus. And if you look below, if you look at the pink structure, that's the nucleolus. And you can see, if you look at now where RP11 is, it's not in the nucleolus. It's all over the nucleus. The green is everywhere. It's like the soldiers who come out of the barracks and are going nuts because they've got something to do, some new job to do. So MLN really changes the localization, and that's contributing to its new function, killing cells. So it's kind of a stress response. Now, one of the experiments that Arthur did in this paper was that he took a version of RP11, this protein, RP11, sorry, that can't be Denedylated. So it's modified and it just cannot be Denedylated anymore. And MLN, of course, blocks Denedylation. And if we put that protein, K0 as they call it, into the cells, you can see that it behaves, it looks exactly like the normal RP11 that's been treated with MLN. So that's the K0 looks as if it's been treated with MLN, but there's no MLN being treated in those cells. We're just blocking the Denedylation a different way. And if you click the next part, so combining this K0 thing, this modified version, with topotecan, kills cells almost as well as MLN plus TPT. And that means that just relocalizing the single protein has a huge part to play in how MLN synergizes with TPT, with topotecan. Okay, so the next slide. So all of that work I just showed you, that's in cultured cells. So what about in a model of retinoblastoma growing in an actual eye? So the first thing you want to know is whether the combination is safe. And I had, thousands of conversations with my collaborator, Jeremy Sivak, who's actually on the call tonight. And I just want to thank Jeremy for joining us. He's a real expert in developing drugs and also using drugs in the eye. And he gave us just tons of advice about this. There's Jeremy. So one of the big questions you always have to address is whether the combination is safe. And so we had to find the highest dose of each drug that's safe, and then we had to combine those doses and ask if that's safe. And we used the mouse eye as a model for this. And the next, and what we found, and we collaborated with another person over at the Western Hospital, Philippe Monnier, to do this, we found that the retina looks perfect with the drug combination, and also, importantly, functions perfectly. So the electrophysiological response of the eye was unaffected by, a combination of MLN plus TP treatment. Okay, so it's safe for the mouse retina. What about if we now treat a tumor? So what we did was we injected human retinoblastomas into the eye, and we treated them with a combination of the drugs. So non means untreated there. So the gray letter, non, is untreated. MLN, the green one, is that all those mice have been treated with MLN. And then TPT is treated with TPT, and MLN plus TPT is the combination. And each of those mice at the starting tumors, that blue blob that you can see inside the eyes, that's actually luminescent. So we've labeled the retinoblastoma cells with a luminescent marker so that we can live image the growth of the tumor inside the mouse eye. And you can see that the untreated on the far left there, after 5 weeks, it's really growing quite big. In 10 weeks, it's even bigger. If you treat with MLN, there's some limiting of the growth, but by 10 weeks, it's quite big. And the same with topotecan, it goes away pretty effectively. There's a bigger army, but it comes back. It can't hold on. If we treat with MLN plus TP, it goes away and it stays away. And those were 1 injection per week for 4 weeks. So the combination is more effective than either single drug, and it can induce these complete responses. So the next slide is just a wee summary of everything I told you. So, some retinoblastoma cells are sensitive to this drug alone. In sensitive cells, the drug engages a really effective killer army, but in resistant cells, very few soldiers are activated. But the resistant cells are sensitive when you combine MLN with a classic standard of care chemotherapy, and that's synergy. And here we showed that there's new soldiers engaged, and many of these are these RP proteins. And MLN totally disrupts the location of these proteins, engaging their killer function. And I also showed you that the combination of MLN and TP doesn't harm the mouse retina structure or the function of the retina. And the combination kills human retinoblastoma tumors grown in a mouse eye more effectively than either treatment alone. So I hope I didn't lose you, and if you have any questions later on, I'm very happy to try and explain a wee bit better. Thank you for listening.
Awesome. Thank you so much, Rod. That was amazing. I'll just give some of my final thoughts. So as a patient, looking at this study, MLN does look very effective. It is preclinical, but it looks very effective in the study itself. And in terms of side effects, which as a patient, this is something that, you're pretty concerned with, it does offer that potential of reducing the chemotherapeutic usage, which reduces side effects. And one thing I do want to point out is when retinoblastoma is treated, when someone is a child, that's not something that's just affecting them at that point. Retinoblastoma is something that someone will live with for their entire life. So one of the things that's always emphasized is, early detection and prompt treatment. And when I look at this as a patient, MLN looks to really help with treating retinoblastoma very promptly. So as I've put in the last point, not only is it a better outcome for retinoblastoma for these patients, it's also a better outcome for the rest of their lives. So thank you guys.
That was fantastic. Both of our presenters you guys did a fantastic job in explaining a very technical topic that's very hard to understand, and that really brought me back to some of my beginning times of being a new retinoblastoma parent and trying to read some research articles, which I just could not get through and did not bother reading further because I just didn't understand. And I think here you both really explained the research at a easy to understand high level. And then Rod, you really helped us understand the more scientific aspect of it and why this is a good idea and how it came to be studied, and what studies were done to better understand it. So I really appreciate you both explaining this topic to us. So I have a list of questions that our audience could get started in thinking about and discussing. I would love to hear from you guys in the audience, your thoughts about the article, about the topic. Maybe somebody with lived experience, maybe could share, when you hear about new RB treatment, what hopes or concerns do you think about? If this is something you would like to share. Researchers, if somebody could comment about how does this drug move to clinical use? We've heard how this was tested, but what's the next step in the process, and what would that look like to perhaps be part of standard of care? If somebody could comment, just picking out a couple of questions. And maybe from a clinical perspective, what makes the study feel promising? Or maybe does anything make you feel cautious and you want to share? If anybody wants to get started. Is there somebody who can comment about maybe drug development and sort of life of developing and moving it to clinical use?
We, I'm not a clinician, so I can't take any part in that. But we did one of the ways. So one of the ways that you can take this forward, if you obviously need clinical partners who are enthusiastic. But one of the ways that it's expensive to run clinical trials, and one of the ways that you can engage a clinical trial is by engaging the company that makes the drug. And so I contacted the company that engaged the drug, and actually Francis Money in Switzerland, who was Arthur's external examiner for his PhD, he was very interested in working with them to maybe try a clinical trial. But the company told me, we've already got a number of cancers. They're mainly focused on leukemias with this drug, and there's some solid cancers, other solid cancers that they're trying, and they didn't want to start any new clinical trials. So immediately you hit a block there with the company. And so where do you get the funds to do this? You can still run a clinical trial, but you need to find the funding and to drive it forward. So there's a whole bunch of new blocks, there's a whole bunch of new problems, and actually maybe Jeremy, you could comment a little bit on these kind of things, because Jeremy used to work at Novartis, which is a big pharmaceutical company.
Hello, can you hear me okay?
Yeah.
Yep.
Yep, great, thanks. Yeah, thanks. I just want to also congratulate Dhruv and Rod for fantastic presentations. It's been really interesting for me to see this format, actually, because this is retinoblastoma is not my field of expertise, but I think it's really wonderful that you have these sessions. Even though I was part of, peripherally part of this study, I learned stuff from the way it was presented by Rod just now. So that's fantastic. I just wanted to comment that the models that Rod's lab uses are incredibly sophisticated, like really cutting edge stuff, pushing the models themselves, like the techniques themselves, as well as the analysis of the biology that's happening here. So that's it's really amazing work to see. And those kinds of models are particularly powerful for what I think would usually be termed pharmacology, or essentially the effects of the drugs on the body, or in this case on the tumors. But there's the reverse side of it, which is often referred to as pharmacokinetics, and that is the effects of the body on the drug. And there's an incredibly complex interplay of things that can happen. So the drug gets broken down and metabolized into different types of molecules. Those themselves can interact with other drugs that a patient is taking. That can have safety concerns that were unexpected, especially when you're talking about taking maybe two drugs and they may interact. So on the one hand, the synergy means it's wonderful, you can lower the dose of each, but you may not fully understand some interactions or some unexpected things that might happen because of both drugs being in your system at the same time. And the point that Rod made about the company that owns this, so I think it was originally developed by a company called Millennium Pharmaceuticals, which is where MLN comes from. And they just name all their compounds after themselves, and I think that was bought by Takeda.
Yeah.
Is that they probably, because this is in the clinic, they have access to all that information. And so without having that information, that presents an additional roadblock to moving things forward. They probably could be coaxed to work with you if they didn't have to fund it. But it represents this this issue about anytime you're going to combine things in a new way, you have to really, I think, consider carefully and study how that new interaction might affect the safety profile in particular.
One of the nice things about intravitreal injections is that the exposure to the entire body is massively reduced. That's another really good aspect of intravitreal injection of drugs for retinoblastoma treatment. Whereas, as Dhruv said, he was treated system wide. He was getting injected into his blood system chemotherapeutics, which can have side effects all over the place. Whereas intravitreal treatment, it's minimal exposure for the systemically usually.
Could I make a comment?
Yes.
We're just across the street from each other, Rod, but we haven't had lunch for a long time. And we've gone a totally different direction with topotecan in the chemoplaque, which we deliver only to the eye, but without an intravitreal injection in the little. Do you know about this, Rod?
I remember you talking about it a few years ago.
Yeah. So the data is quite spectacular for its success in retinoblastoma, and that's one point. But it might be a really good way to deliver to the eye over a sustained period of time, because the beacon is in the chemoplaque and it stays there delivering to the eye for 6 weeks. And then we take it out and there's a little pearl of topotecan, which is yellow, left, and that's all. But it has really, the only toxicity is inflammation. Is there any detail on just the MLN? Does it cause inflammation all by itself without topotecan, without a drug, or not? Or you don't know because you're only doing mice eyes?
We have no idea in human eyes.
No.
I mean, systemically, MLN will have some side effects on like blood, certain blood cells, the usual kind of thing. So there's a lot of clinical trials on MLN happening right now, mainly for leukemias, but for some solid tumors as well. So a lot of those will reveal what kind of side effects happen. But that's from IV injection and intravenous, i.e. to the blood system. So those wouldn't apply to. But what MLN does in a human eye, obviously, that needs to be tested. But it had no effect on the mouse retina at all, including electrophysiological, which hardly anybody ever does with new drugs. So that was encouraging.
But a different eye, a larger eye, like a rabbit eye, does it do anything in that context?
We haven't looked at that.
So I think we should talk about the chemoplaque approach, but that's not the second thing that we've done in our study, which now we're at the final stages of writing up this paper, is to design a whole new way to do clinical research for retinoblastoma, which actually becomes very much cheaper and relies on comparing the patients who become research subjects. They volunteer and sign a consent to be in the study. So they are participants. And then we compare them to patients retrospectively who were very similar by all sorts of criteria, which are really easily to work out. to match them, and we can match them, and then we compare the outcomes of how many other treatments they needed to have to cure their cancer in the eye. And the result is quite dramatically different. So that is a much cheaper approach than the normal clinical trial. And I just wonder if we got together, could we go back to that drug company and get them to really think out of the box how powerful this could be for retinoblastoma?
I mean, I think obtaining the drug and doing the trial, if you have these much more economical ways to do it, that's really exciting. So might not even need them. Just get a hold of the drug and do it.
How do you get hold of the drug without the company?
Yeah, Jeremy, you asked Jeremy that question.
Jeremy,
Oh,
how do you get the
sorry.
drug?
Yeah. So I think that so I think I helped Rod with this in a previous context, different company before, and I've actually been involved in other groups that have tried to do the same thing. I think when the cause is good and the argument, the science is solid, and you're not asking for the company to invest in the trial per se themselves, but you just need the material. So one of the big costs at the beginning in terms of putting together a trial is actually that you have to have special super clean clinical grade compound drug. You can't
Yeah.
just use the stuff that would be available for laboratory use.
Yeah.
There's actually very strict regulations for obvious reasons about that.
Mhm.
And so often when a company is getting ready for other trials, especially systemic delivery of the drugs for maybe other cancers in this case, they've probably made a lot of it, and I mean like grams or even kilograms of it.
Mhm.
And so at a very at this clinical grade level,
Mhm.
so getting them, making the pitch to them, understanding the importance of the cause. I would think that maybe just an appeal to try to get them to donate some of that material would have a huge impact in terms um, of letting the trial move forward. They would be nominally involved, but not really very much. All they would have to do is release some of this material that they may not be doing that much with anyways.
Mhm.
And I feel like that's at least a good starting point.
And I think with patient advocacy as well, that might be a good approach too, Brenda.
Yes, yes. So Rod and Jeremy and Dhruv, let's get together
Sure.
in the next weeks, and I'll actually send you some information or some presentations you can put through to see, which won't be up to date. Because the data is being refined down to great details now with the publication. So let's talk about it and move forward on that. This is a very exciting outcome of the cup of tea, I would say.
Absolutely.
Perfect. Thanks, Brenda.
Thanks, Savannah.
Well, if there's any way this recording could be useful to any of that,
It will be, yes. That's
perfect.
great.
Because we summarized, well, maybe that's not the right demographic, but we have summarized the information from our presenters very nicely for anyone to understand.
Mhm.
And then with Dhruv's excellent explanation of why this is important for the lived experience, I think it's a great package. Helen?
Thanks, Stephen, and thanks, everyone. This was really informative. In our last cup of tea, we talked about our biobank, and one of the discussion questions was around sharing our biobank specimens with private companies. So, like companies that, such as the one that made the drug you're talking about tonight. And we find that in the participants in our biobank, not everyone checks the little box that says, yes, I'd like to share my cells, or if we make cell lines with those companies. Now, here you have a really great study that was able to be done in a university setting, and I'm sure you use some of these cell lines like similar to the ones we have in our biobank. Um, we might have some limitations about sharing with a company like this if they wanted to do some of their own studies. So maybe Jeremy, having come from industry as well, could you tell us a little bit about how patients might be involved? It's sort of a new area for our patient advocacy group to work with industry partners. We haven't worked with them that much, and I think we have some trust building to do. We're learning, and some of our members of our patient advocate committee are here tonight. But what would you suggest are good steps? Like all the discussion we're talking about right now, it's very science-based, but what are some ways that our patient group could get to know how these companies work and build those relationships such that we're asking the right questions? Things have come up about intellectual property, profits, who owns the data? Will this help retinoblastoma patients around the world or just the rich ones? So, what should we be asking when we're working with these companies, or how can we learn more?
Uh, that, I can't definitively answer that. I mean, I left industry a long time ago, because in part some of the frustrations that you referred to. But on the whole, I would say that on the scientific side, the people that I worked with were wonderful, altruistic, scientific people who are interested in more practical questions. I will say that I think in the last, decade or two, a lot of the bigger companies, at least, have realized and started to really recognize the value in patient outreach and advocacy, and sort of opening themselves up in ways that they might have been more restrictive of in the past. And this really, I think, represents an internal battle between their own lawyers and sort of intellectual property advocates and their scientific staff and clinical efforts. And so a lot of companies have kind of created a structure to allow for those kinds of collaborations. And I don't know how one would do this, to be honest, at all, but I would think that sort of inquiring in that direction, and kind of wondering whether there is a structure within the company to allow for academic, for example, collaborations might be a way in, that would allow for the sharing of reagents and materials, or what the company would consider it donating. That might be a way to sort of leverage them and engage them with patients and advocacy groups that in the past they just wouldn't have been interested in. That's sort of what comes to my mind immediately. But I'm certainly not an insider anymore in any way, and so I don't really have anything, beyond that. But I do feel like the contacts that I have maintained in industry are more open and have mentioned these kinds of avenues much more often recently than they used to in the past.
Thank you.
Yeah.
I
And I just want
actually,
to apologize and say I have to leave to go to another engagement, but I really, again, thought this was a wonderful group. And if anyone else wants to reach out to me with more of these kinds of questions, I'm happy to answer. And to Brenda's point, I'm very happy to be part of this conversation if you think it would help going forward.
Thank you so much, Jeremy. It was
Thank you
great to
all.
hear from you.
Thanks, Jeremy.
Thanks, Rod.
Thank you.
could I just comment on what Helen's just talked about and the difficulty in working across with industry, et cetera? Um, there's a whole new movement coming. Maybe this is one of the good things of the attack on science in the US, because the industry parts come up as being a valid way to do your science instead of being that kind of out there just money grabbing kind of attitude that we grew up with, where we had to be academic and have our criteria, our careers depending on qualities of journals we published in, et cetera. That whole thing has shifted a lot, particularly with the money it goes to the industries. But even in the Gairdner Awards, there were two examples of fantastic industry partnership in building what the award was placed for the great discoveries. And actually, I can think of a third too, that where industry really backed fully was a full ethical partner in the science. not that enemy out there. In fact, made it all happen. In fact, one of the Nobel Prize winners this year is a woman who did all her discoveries that came to the Nobel Prize in a company. And so it's really a changing world for the better, so that the money, which is in the industries, can be supporting the work that Rod Bremner and Dhruv are doing.
Thanks, Brenda.
Good point.
That's
But
great.
there's some publications coming out you can see on this, which are quite fascinating.
That's great discussion tonight. We have just wrapped up here. We're 1 minute left. I wanted to leave our last minute for anyone who has lived experience, if they wanted to comment about anything or ask a question.
I can go. So I think what everyone's brought up is amazing. I think in terms of reaching out to the company, I would be more than happy to help with that. Please let me know if there's anything I can do. I'm again not too familiar with it. Um, but yeah, I just like to thank everyone for being here and yeah, I think it was awesome.
Great. Thank you, guys. I just want to thank everyone for being a part of our cup of tea. If you have any questions about today's presentation or any opportunities to partner in research, you can email retinoblastoma.research@sickkids.ca. And I'm reposting those links from the announcements I shared earlier. Please fill out our survey for the cup of tea topics. The more people we have feedback on, we can really pick the topics that are important for our community to hear. You can follow us on social media and visit our website, rbcanadaresearch.com. Thank you, everyone.